Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we'll be talking about picking OTUs. OTU stands for Operational Taxonomic Unit, so this is basically just a cluster of sequences that we think are closely related. So what we're starting with here are the amplicon sequences. So this is not as often something you would do with shotgun data. So it's basically the same gene over and over again with subtle variations, and we're trying to group sequences that are very similar into bins. So why are we doing this? Well, for one thing, it helps us go from the millions or hundreds of millions of input sequences down to a small number of usable features. The reason we can do that is because we're controlling for various types of error, so sequencing error, error that came from the amplification steps, PCR error. And those aren't real biological errors. So if we have two sequences that are different because of sequencing error, we don't want to consider them separate species. We want to group them into the same bin and count them as one feature. And we're also controlling for very recent evolutionary divergence. And this is a much more debatable part of the process, which is discussed in detail in another video. So here's some terminology for OTU picking. These are the high-level approaches that people use for doing this type of clustering. You can simply cluster your input sequences without regard to any known sequences. So this is called de novo clustering, or picking de novo OTUs. And it's just a big machine learning or data mining problem. You know, how do you identify the best clusters? And then the complementary approach is closed reference OTU picking. So this is where you simply take every input sequence, find a close match in a reference database, and then you call it that OTU or that species. And here, if anything doesn't hit the reference database, you throw it out. Then finally, you can use a hybrid approach, which is referred to as open reference OTU picking. And this is where you first search everything against the database, and then anything that doesn't hit, you do de novo clustering there. So open reference could be more useful if you have samples, say, from non-human primate gut, where a large fraction of the species that are in there will be represented in the reference database, but there's another whole fraction of the community, you know, say 20 or 30%, that we don't have examples of in the reference database, and you don't want to just throw that data away. The first type of clustering that we'll talk about is centroid-based. This is a common approach in OTU picking. It's used by Uclust, which we'll talk about later. And the requirement here is that within each cluster of sequences, when you pick a single centroid sequence, all of the rest of the sequences should be within a certain amount of variation of that centroid. And then you also want other clusters to be clearly separate. And the rule that you can use here is to require that the centroid of another cluster is far enough away from the centroid of the first cluster that they wouldn't be merged together. So that doesn't say anything about the space in between the clusters, in between the boundaries of the clusters, because you could have two sequences in two different clusters that are actually quite close to one another. The next common approach is single linkage clustering. And all of these clustering approaches are used in data mining, so they're not just for OTU picking. Single linkage is sometimes called a friend of a friend clustering. So to be in the red cluster as a sequence, you only have to be sufficiently close to at least one other red sequence. And so these can tend to have long tendrils and kind of expand out through this friend of a friend of a friend approach. And it also doesn't say much about the boundaries between the clusters. And so you could potentially have clusters that are quite close to each other, as in the picture. Another common approach, complete linkage clustering. So complete linkage is where everyone in the cluster has to be friends, so they all have to be sufficiently similar to one another. These tend to be more spherical in shape. And finally, average linkage. So average linkage is sort of a compromise between single linkage and complete linkage, where you're simply requiring that the average distance within a cluster between all of its sequences is above a certain threshold. What do good clusters look like? So that describes valid clusters. How do you know if a clustering is actually a good one? Because for a given data set, you could actually have many different valid clusterings. And the general rule is you want, within clusters, you want the distance to be small, so you want the clusters to be tight. And then you want the distance between clusters to be large. And ideally, you want to have a large space even between the boundaries of the clusters. So, the smallest distance between any 2 points in these two clusters is still fairly large. Now, when we're doing OTU picking, de novo OTU picking, there are certain advantages and certain disadvantages. So, one nice thing about de novo OTU picking is that you're using all of the sequences, so you don't have to throw some out, as in the closed reference OTU picking. And this allows you to study unknown species. So if there's a certain type of sequence that's showing up lots of times in your data, it doesn't match anything in the reference database, it's the fact that it's showing up lots of times means it could be some new species, and often is. On the other hand, most of the de novo OTU picking approaches, these clustering approaches, are not easily parallelizable, so they tend to be very slow. It's often the bottleneck for doing analysis of microbiome data. And they don't generalize from one study to the next. So if I pick de novo OTUs in my data set and you pick de novo OTUs in your data set, then we can't compare our OTUs. So we'd have to combine our data and then cluster the sequences. Now, I'm going to talk about a number of the most common and most commonly used de novo clustering approaches. Possibly the most common is Uclust, and this was developed by Robert Edgar, published in 2010, and it's actually a fairly simple algorithm. So it does greedy clustering. What does this mean? Greedy means you're kind of building the model as you go and hoping it works out. You're not taking a step back at any point during the process and reevaluating what you've already done. So the way we do this is you open up the sequence file, you pick the very first sequence, and you call that a centroid. So already, that's obviously wrong, because what are the chances that that sequence is going to be a good centroid for a cluster? Then you pull out the next sequence. If that sequence is sufficiently close to the first sequence, so if it's within 97% similarity, then you add it to the first cluster. If it's below that threshold, you use it as a new centroid. Then you pull out another sequence, and you compare it to both of the first two clusters. If it's sufficiently close to either one, you add it. You just pick one. Hopefully you pick the closest. And then and you move on. If not, you make a third cluster. And you just keep doing that through until you're done with all of the sequences. So to make this work, you have to have a way of comparing one sequence to another. You have to measure that percent similarity. And that is not trivial. So that requires aligning the two sequences, and it's very time consuming because there could be gaps inserted on either of the sequences in any position. So it's computationally very intensive. What makes Uclust fast is that it uses usearch for comparing the sequences, and usearch is a little trick that it basically looks for short words. It characterizes all of the short words in each sequence. So say every 8 bases might make a word. and it asks how much overlap there is between the words in this sequence and that sequence. And that's a very fast thing to compute. You don't have to worry about gaps. So, so you take a new sequence that you haven't compared to anything yet. You look at its k-mers. You see what the overlap is between its k-mers and the k-mers in each of the centroids you've already picked. And you use that to make a guess about which ones it might match closely. Then you take the top 32 or so, and then you do actual alignment where you find out what the percent identity is by doing a local sequence alignment or a semi-global sequence alignment using the Smith-Waterman algorithm. So the challenge here is that and so then with the Uclust approach, you usually try a certain number of matches, and if nothing hits, you just assume there's no hit. But that's based on the k-mer overlap, so it doesn't mean that you will always find a hit if there's one in there. So problems with this. Well, number one, it's closed source. The algorithms are described sufficiently well to understand what they're doing in general, but we don't know what the exact implementation is. You also have to pay for it if you want to use it on big data sets. The order of sequences matters a lot here. So the very first sequence becomes the first OTU centroid. And if that happens to be a bad choice as a centroid, it can kind of throw off a lot of your clustering. One way around this is to just sort the sequences by how common they are. So if you have the exact same sequence showing up thousands of times, it's probably a decent centroid. So because of the U-search comparison, this guessing what the best matches might be for a new sequence, you are likely to sometimes miss the best match. So a sequence may not go to the best cluster. And you also may miss a match entirely. So a sequence may in fact match something in the current OTU clusters at 97%, but we didn't find it because we're doing this approximate approach. So this means that you can make too many clusters, and sequences won't always go to the best cluster. Next method, CD hit, is it was the first major player in this space. Actually, it was published in 2001 originally for clustering protein sequences. It's almost identical to Uclust, except it doesn't use the fancy trick for comparing sequences to each other, where you use the overlap in k-mers. So it's a lot slower. Edgar claims that Edgar's the publisher, the author of Uclust. He claims Uclust is better, produces better clusters, but there hasn't really been a rigorous comparison. Next, Sumaclust. So Sumaclust is also basically exactly the same as Uclust, but here it's using exact alignment or optimal alignment at each of the comparison steps. So rather than using the k-mer-based guess for which matches, which sequences might be a match. And so this might mean that you have less false splitting of clusters, because if there is a match for a new sequence, you'll find it and you'll put it into that cluster. And it also means that you probably improve the assignments to the best cluster. So each sequence goes to the correct cluster or the closest cluster. However, it's going to be a lot slower than Uclust, and there's also no detailed published reference. So there's an abstract for Sumaclust, but no publication at the time of recording. There's also OTU capability, clustering capability in the Mother package, very popular package for analyzing microbiome data. Mother is interesting because it actually uses all of the pairwise distances between the sequences. So this is a challenge because you have to generate those pairwise distances. You could do that using various approaches. You could do that using Usearch, but then it's got the approximation issue. You could use it doing full optimal alignment, but that's going to take an insane amount of time. However, once you've got the distance matrix, you can start to do real single linkage or complete linkage or average linkage clustering. And that's actually what Mother does. But they call these single, they call nearest neighbor, complete they call full neighbor, and average linkage they call average neighbor. So pros for Mother, it has this kind of bird's eye view of the clustering. And so this is generally going to be better than a greedy approach. And then a con is that you have to have this full pairwise distance matrix for all of your input sequences, so this is not really feasible when you have big data sets, depending on how big it is and how much RAM you have. So the last method I'm going to talk about for de novo clustering of sequences is swarm. And swarm is very interesting. This is currently published as a preprint, so it's not, it's a preprint, so it's not peer-reviewed, but has a really intriguing approach. So the approach is actually quite simple. What they do is they choose an arbitrary seed for a new cluster, so same as Uclust. But then rather than walking through the rest of the input sequences to find out if they should go in the same cluster or be split off, they check that arbitrary seed against the rest of them, and they take all of the sequences that are within a very small threshold, like say 1 nucleotide difference. And that's the D that you're seeing in this figure. So now that cluster has gone from 1 seed to that seed plus a bunch of subseeds that are very close to it. So in figure B here, you can see the sequence at the center would be the seed. Then there's one step where you add a bunch of subseeds around it. Then they repeat. So you take all of those subseeds and look for anything in the reference database that's within D distance, or like 1 nucleotide mismatch, for example. And then they make another ring around the cluster, and then they take all of those sequences, add them to the cluster, and search for sequences that are within D distance of them, and they just keep doing that until it stops growing. So the major benefit of this is that it doesn't really matter, because you're using this kind of single linkage clustering, it doesn't really matter which sequence you start off with as a centroid. Because in the end, the cluster will be defined by single linkage, not by distances to the centroid. So it takes care of the arbitrariness of choosing centroids. In addition to that, you won't, because it's single linkage, you won't get this case that's shown on the left side where you can have centroids in one cluster that are very close to another cluster. But they happen to fall just outside the threshold for being included in that cluster. So you end up with basically false clusters, like too many small clusters. One issue with swarm is that it's assuming that there are big gaps between the OTUs, that there's not just kind of continuous sequence variation that bleeds together. This is a major issue when you're dealing with really short reads from amplicon data, because you'll have species that, 2 different species from the same genus that have identical sequences. And then there are lots of sequences that might be off by 1. It becomes less of a problem as you're getting longer and longer reads. They have a method, so that would look like this picture here, where you really do have different species at some point, but their marker gene sequences are too close, so they're almost kind of blending together. And so the authors claim to be using the internal structure of OTUs to fix this. So after they do that single linkage clustering, they go back and do some kind of splitting up of the clusters. And I'm not sure exactly what that is, but it probably works reasonably well. So that's the end of de novo OTU picking. And de novo OTU picking is actually extremely important because this is what generates the input data for the rest of all of our analyses. If you don't end up with good features, you're not going to get good biological signals. So we call that garbage in, garbage out. Now, close-referenced OTU picking. So for many of you is going to be the easiest and most sensible thing to do, especially if you're studying the human gut or maybe human oral communities, any community where we've already characterized the bugs in that community very well. So if you're studying the human gut, there's no sense most of the time in doing de novo clustering on all of the sequences because we can just compare them to a database of known sequences. And it's also really fast, it's really easy to parallelize. So this is actually what I do for most data sets that I'm analyzing. And in a typical human gut sample, you can get maybe 95% of the sequences to match something in a reference database. So benefits of closed reference OTU picking has built-in quality filter. So if you have erroneous sequences because of sequencing error or PCR error, they're not going to hit the reference database. It's also, as I said, easily parallelizable. The OTUs can be defined carefully using curated sequences, highly trusted sequences in theory. And of course, the reference database is getting bigger and bigger all the time. So at a certain point, for most environments, we shouldn't really need to be doing de novo clustering. And that's why I'm investing heavily in the reference-based approaches. Cons are, of course, anything that doesn't hit the reference database, you throw it out. So if you try to do reference-based OTU picking or OTU clustering in a soil sample, for example, you're going to be throwing out half your data because the bugs just aren't in the reference database. And of course, another disadvantage is that you're still susceptible to errors in the reference database, which we know are there. Here's a nice little plot from the Earth Microbiome project showing what fraction of sequences tend to hit or tend to be missing from a reference database depending on the environment. So lower is better because you want most of your sequences, you don't want a lot of sequences missing the reference database. And so all the way on the left end is the human gut microbiome. And in that case, this plot shows there's maybe 10% of sequences might fail to match the reference database. But all the way on the other end is marine habitat, river, creek, rock. And these are cases where 60, 70, 80% of the sequences may be completely outside the reference database. So it's really only on, at least for now, it's really only on the left end where you're doing host-associated sequences that you should be using a strictly reference-based approach. The last approach is open reference OTU picking. This is generally favorable to de novo picking because in a lot of environments, a good fraction, maybe half of your sequences will hit the reference database. And so the nice thing about this is that the reference-based part of open reference is really fast, but then you don't have to throw out your data. It's partially parallelizable, but that means you still have to do the de novo part, and that's still really slow in big data sets. And you can also have an issue where you have two different quality levels of OTUs. So the reference-based OTUs might be better quality, and depending on your clustering method, the de novo OTUs might be more prone to sequencing error. So in summary, if you're trying to choose an OTU picking approach, I would use closed reference picking if you're dealing with human or mouse samples from the gut, skin, oral, nose, because these are well characterized. I would do de novo picking if you have really uncharacterized environments like soil samples or new water samples. Also, you need it for building the reference-based OTUs. And then finally, I would use open reference picking for everything else.